welcome to Learn Extra Live. Guys, I missed you so much. Today we're with Kathy. Kathy, how are you?
Fine, I missed you.
I missed you too.
I have you and Kai, it's sort of like alternating weeks,
Hey.
yeah.
I know, and I've missed you guys too, more than Kathy even.
Oh,
Trust
thank
me.
you.
Just joking, guys. I hope you are ready for a fantastic show. It's life science today. You guys know when you see Kathy, you see life science. So today is a very exciting show. Last week, remember that you did the structure of the dicot root. This week, we'll be doing the structure of the dicot stem. So it's very exciting, and it's like a nice follow-up. So you guys can take out your notes from last week, bring them on to this week so that we can go through everything. Hey, Kathy?
She's far too excited about dicot roots and stems, guys. She clearly isn't sitting in class with you at the moment, but we're going to try. We're going to make it grow. How's that, eh?
Yes, yes. And I know you guys are excited. Don't lie to me. Guys, we'll be going for a quick break. Make sure that you start chatting to us on Facebook. Remember, our Facebook account is facebook.com/LearnExtra and Twitter @LearnExtra. See you after the break, guys. Bye. Welcome back, Mindsetters, to Life Science with Kathy. I know you guys are excited and have your pens, papers, books, everything in color pens ready for a fantastic show. Remember to interact with us on Facebook, guys. Ask anything and everything on the show because that's what we're here for. We're here to answer your queries and concerns. So when this show ends, you guys are life science buffs. Kathy, we're so ready for you. Please take it away.
Thanks, Kat. Okay, people. Last week, we did the whole structure versus function of a dicot root. All right, remember, we're looking at dicot plants. Dicot plants are part of the family of angiosperm. So we have angiosperm, which is flowering plants, and there are two types of angiosperms. We have monocotyledonous and dicotyledonous. I put that table up last week for you to look at, where we had a comparison between the roots, the stems, and the leaves. We looked at the roots last week specifically. We are now looking at the stems. All right. We're looking at the cells and the function of the cells within the stem. Now, epidermal cells, you know, are there to protect. Okay, and we have various modifications to our epidermal cells, which you have learnt about. For example, in the root hair, that's an epidermal cell, and it's an extension in that epidermal cell. But your epidermal cells are one layer, and they are there to protect. All right, so now we're going to look at the stem. I'm going to look at all the different types of cells again. We're going to do epidermal cells, we're going to do the cortex with the parenchyma cells, endodermis, then we're going to do the vascular bundles, all right? Now remember, in the root, it was in the shape of a cross, all right? So the xylem was in the shape of a cross with a phloem in between. Here, we're going to have actual vascular bundles, and they are in a little circle, and that's important for what we call secondary thickening, which is when we get stems that have bark on them. In other words, they're big and old, etc., etc. Okay, so all you have to do is remember what the different cells' functions are. And if you do that, you can't go wrong, because now we're going to look at the cells and their function in the stem specifically. All right, we're looking at the dicot stem and the functions of the stem. Now, we know the functions of the stem, where the roots was to anchor and absorb. Here we have the fact that it provides support, all right? What else does it do? It is a transport mechanism. Let's just... a transport mechanism for water and dissolved nutrients. It transports the water up the plant, and it transports food and water up and down. So there we go. All right, and here it is only up, so from the roots all the way up, and it serves as storage organs. All right, so if we look at the roots, what did they do, people? And this is really important. Remember, a root anchors the plant. It absorbs water and mineral salts. That's why we have the root hairs. very important that 80% of the water is absorbed in that little root hair region on a root. Okay, and what else did it do? It was a storage organ. Think about it. We eat carrots. What is a carrot? Nothing but a big taproot, all right? And we eat them. So people, please remember it's also a storage organ. With the stem, here, it doesn't anchor, but it supports. Because if it isn't for the stem, where are the leaves and the flowers and the fruit going to sit, okay? It transports the water and dissolved mineral salts up via the xylem, the food and the water up and down via the phloem, and then a storage organ, because in some cases we eat the stems. And all you have to do is think about what a celery, we have the leaves at the end, but those leaves sit on top of and are attached to that stalk, and the stalk is actually the stem. So that just came to my mind in the first place. So we do eat the stems, but in big trees we don't eat the stems because, well, they're too huge to eat. But they transport water and mineral salts, number one. They transport nutrients up and down. Number two, they provide support for the leaves, etc. Number three, and number four, they store food and nutrients for the actual tree itself. Okay, now the structure of a dicot stem, and this is what it looks like on the inside. Okay. First of all, you're going to have hair-like outgrowths. Now, what is important about those hair-like outgrowths is that they are multicellular. Okay? They're multicellular. In other words, they are different from a root hair. Where your root hair was unicellular. So if we had the root hair, we had an epidermal cell, another little epidermal cell, and then this extension which made the root hair. And then you'd have another epidermal cell. So your root hair is unicellular. Whereas here, we're looking at a multicellular. So we would have the epidermal cell, and we would have another one and another one. So it looks similar to what I drew there. And then we'd have little outgrowths, which would be multicellular. Very important, people, it is multicellular, not like the root hair, so it's different. And that is one of the ways you'll be able to tell whether the diagram that you're given is a root or a stem. If it's a root, the extension out of the side in a cross-section is going to be unicellular. If it's multicellular, it's a stem. That's one of the differences. Also, you have your epidermis, and then you have your collenchyma and your parenchyma and a starch sheath, which makes up the cortex. Then you have medullary rays. Now, if you look here as well, at the, and you're not going to be able to see this, so let's get another color here. There are the vascular bundles. All right? And those vascular bundles in the root are going to look like, I'm going to draw it here quickly. You'll have, if that's inside the endodermis, that's what the xylem will be, and the phloem will be little areas in between. So if it's got like an X in the middle, and I mean, I know this sounds very simple, and it sounds like, no, come on, use biological terms, but it's all fair and well using biological terms, that you need to be able to tell the difference between the two and not get them confused. So if it's got a little X and it's got the little sort of little circular type structures on the sides here, there's your phloem and there are your xylem vessels. Here you have separate little vascular bundles, and that is what's important, so that you can actually see them. All right, now, the medullary rays are just the way these little vascular bundles are laid out. Then, if we look here, right in the center is a sort of an area that doesn't really, it's got lots of air spaces. And those air spaces in there make up the pith, which is the center part of a stem. Then you have your sclerenchyma cap, which sits just here on the outside around each, that cap. It forms a cap, I think is the nicest way to describe it. A little cap here on each vascular bundle. And that sclerenchyma cap is for strength. Because remember, sclerenchyma is strengthening tissue. So wherever you see sclerenchyma, it's there to give shape and to strengthen and support. So it's on the outside, it's like a little cap around each little vascular bundle. Now the vascular bundle itself is made up of xylem in the center, and let's get another color here. So we'll have xylem in the middle. Okay, now if you look at the root, the xylem was in the middle. So here, the xylem is still in the middle, all right? Always in the middle. And then you have cambium. Now let's get another color here. Let's use green. And we have the cambium. This is nice, it's like coloring in. And then we have the phloem, and here we're going to have a slight little problem. We're going to go with yellow. So we'll put the yellow there. Oh, no, 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 no. I did that completely wrong. Let's leave that one. And we've got, so the yellow is the phloem. All right, so yellow is the phloem, the xylem is the green, and the cambium, the red and the cambium is the green. That makes up the vascular bundle. Now, what is a vascular bundle? Its job is to transport water. Okay, it's a very, very set structure that has on the outside the little sclerenchyma cap, then it has the phloem, then it has cambium, and then it has xylem. Now, with the cambium sitting in the middle, all right, so we've got our cambium over there, sitting right in the middle, and we've got the yellow on white. Okay, let's do that, and then I can go back to yellow because you can't see it otherwise. Okay, there. With the cambium, it is, or contains, I'm going to write it down here so I've got space, meristematic tissue. Why? Because it creates new xylem and new phloem. And as it makes new xylem, the old xylem is pushed inwards, and the new xylem is, the old xylem is pushed inwards, and the old phloem is pushed outwards. And as it does that, so as it makes new cell layers, it's pushing out that way. So it pushes towards the center, and it pushes outwards. And that is what's going to give the secondary thickening, which is what we see when we look at wood. All right, so you must know this. This is very, very important. This is an absolutely NB diagram. You must know this diagram, please, people. All right, now, moving along. What I've got here is what a young cotyledonous stem looks like. Okay, let me get green here so that we can write on here. All right, there, they've stained these cells, and you can see your vascular bundle. So there is your vascular bundle. And what are you going to see? Over here is the sclerenchyma cap. All right, then you've got this region over here, and that little region is going to be your phloem. And then the cambium, and this here, can you see all the thickening on the sides? Nice big thickened cell walls, and they're thickened with lignin, and there's your xylem. All right? Now, looking at this, you can actually see a layer here all the way around. That is the endodermis. Okay, let's go. I'll redo this. Okay, there's the sclerenchyma cap. Then the next one is the phloem. Then we have the cambium. And then we have the xylem. All right? I hope you can see this. There's the xylem. All right? And then we have the endodermis going all the way around here. And right in the center, you can see the spaces are quite large here. This here is the pith. Right, so all your little vascular bundles are arranged along the outside. If they give this to you, you need to be able to label it and talk to it. Okay, let's go back to our green. So, right. What is the epidermis's function? People, I simply put this in because Lindsay here at Mindset is loading up smart, what did they call it? A smart set, smart notes for you, which is all the PowerPoints that I've done for you to learn from. Okay, so that's why I've put it there. But use your brain. They're brick-shaped, they thick cell walls, they have no intercellular air spaces because remember, your epidermal cells look like that. They're just sort of like these square little, let's do it like that. There we go. They're solid little cells. Okay? They have a waxy, waterproof cuticle if they are young. So when your stems are young, or they are, it's before they start their first year of secondary thickening, which is when they'll start making cork cells, which is what makes the bark, and that sort of brownish color that we associate with the bark of trees. Before it gets to that, it's still a young plant. Those stems are going to be green and they are going to have a waxy cuticle around them. Why? Because on the root, we didn't want a waxy cuticle because we want the water to be absorbed. And if it's a waxy cuticle, it'll be waterproof. But on the stem, we don't want excess water loss. So there is a waxy cuticle outside of the epidermis, so it'll sit there like that, outside of the epidermal cells. All right, then the woody stems, the cells below the epidermis, form cork cambium. Now remember, cambium tissue is meristematic. It makes new cells. So that's where your cork cells will be made. So what is the function of the epidermis? Number one, protection, and number two, to breathe for gaseous exchange. Now, stomata, when we did the structure of a dicot leaf, when we did the plant organ, the dicot leaf, you did the stomata. Now, the stomata were those two bean-shaped structures that we found in the leaf with a thick inner wall and a thinner outer wall. And what they did was during the day when photosynthesis took place, because the stomata contained chloroplasts, they would photosynthesize, they would produce glucose, that would pull water into the guard cells, and it would open the pore because these inner walls are thicker than the outer walls. Now we have that on lovely little green stems, but as the stems now form their cork cells, and they now, because of that they're getting older, it's only in the young green stems that we are going to find a waxy cuticle, number one, and number two, stomata. But as they get older and the cork cambium forms, what do we have? We can't have stomata, so the woody stems have a structure called a lenticel. And lenticels are like little holes. So just going back here, function of the stem, in the epidermis in the stem is protection and gaseous exchange. Now, if we go here, I want to show you this. Have a look. There are your lenticels. Can you see them? So this is as the stem has now got older, And the cork cells are now starting to, the cork cambium starting to produce cork cells. All right? What are we going to have? No more stomata. The stomata now develop into lenticels, which are therefore gaseous exchange, the breathing that the plant has to do. Cats?
I think we're ready for a break.
I think so too.
That was a lot, guys. I hope you guys are taking it all in because you're so energetic after school. And guys, hope you also have a fantastic break. Get re-energized because there's going to be so much more exciting stuff. See you after the break. Welcome back, Mindsetters, from your break. I wanted to remind you that remember last week on Kathy's show, we reached the 10,000 likes stuff. It was awesome. And I wasn't here for that, but today I am. And I think that we should start another 10,000 just to get rolling again. So guys, keep liking on the page, like each other's comments and questions and statuses and post anything you like because we're going to like today and we're going to like a lot because we're on Kathy's show and we like her as well. So Kathy, over to you.
So sweet. I'm going to take her home with me. All right, people. This is what it looks like from the outside. Those are the lenticels. All right. They are breathing holes. They get for gaseous exchange. Now, if we look here, this is what it looks like under a microscope. So this here would be your lenticel. All right, that's what it would look like. It's almost a burst in the epidermal layer. Okay, that was just to show you quickly the cortex. What does the cortex do? Now remember, we've got 3 types of cells in the cortex. Okay, there's collenchyma, there's parenchyma, and there's a starch sheath. Now, think of it logically. Collenchyma, what is it? It has pectin and cellulose thickened cell walls. Okay, why would it be there? It's there for strengthening. Okay, because that pectin and cellulose in the cell walls and the corners that start to get thicker, it's there to provide structure and strength and support within the center of the stem. Okay, or in the stem. So that's very, very important. But this, if you just think back when we did a plant tissue, and I said to you, this is what sclerenchyma does, this is what collenchyma does, this is what parenchyma does, you need to know that. And if you learnt it when I told you to, here, you're just simply applying it to the structure that it's in, whether it's the roots, the stems, or the leaves. Right, now, parenchyma. What is parenchyma's issue? It has intercellular air spaces. Why? Because it stores water and nutrients, and it has to have those intercellular air spaces for gaseous exchange. So, what is the function of the parenchyma in the stem? Well, in a really young green stem, you will find that you'll have, which one? Which kind of parenchyma? It's going to be collenchyma. Why? Because it contains chloroplasts. That's what makes a little young stem green. Here, as it starts to get older, what is the parenchyma's main function? Come on, guys. To help move water and to help move food and to be able to store food. All right, so, and also gases. If these cells don't get oxygen, they die. So that's important. There must be gases exchanging throughout the stem all the time. And then the starch sheath. Now, the starch sheath, very important. Why? Because you've got a tightly packed, thin-walled cells. And what is their function? They've got no chloroplasts, but they contain the starch grains or the starch granules. Okay? And those starch granules allows the movement of the water and the nutrients from the vascular bundles to the cortical cells. So that's what they are there for. It's, again, a kind of a storage organ. And remember, plants will store glucose as starch. Okay. Why? If you go back to when we did all those organic compounds and nutrients, we store glucose as glycogen in our muscles and our liver cells, but a plant stores glucose as starch. Okay, and if you look at a starch molecule, it's just a whole bunch of glucose molecules joined together in a polymer, in other words, in a big molecule. Why does it store the starch or the glucose as starch? Because remember, glucose lowers the water potential so that osmosis can take place. But with starch, it's osmotically inactive. It doesn't allow movement or it doesn't attract water to starch. Why? It doesn't lower the water potential. All right, sclerenchyma cells. Why would we have sclerenchyma? Remember, very thick cell walls. They're thickened with lignin and they become waterproof. And because they're waterproof, the cell contents are dead. And I did put it here. So it forms a lumen, which is that central cavity. Okay, because remember the cell walls are all nice and thick like that. Okay, so the lumen is quite small and you have all the contents of the cell will be dead. Why? Because this lignin is completely waterproof, so nothing can get in there, so the little cell dies. Okay, the sclerenchyma fibers are long and tapered ends, and what do? They help the plant to be flexible, but they provide strength and also protection of the vascular steel. Now remember, your sclerenchyma forms the little sclerenchyma cap. Okay, that little cap, and that is the strengthening and protection that we're talking about here of the vascular steel. The vascular steel is made up of the xylem and the phloem and the cambium, which is in between. And then we have the sclerenchyma fibers. What do we do with them? What is their function? Well, we make ropes, and you've done this. I know you've done this because I remember doing it with you. It's used to make rope, it's used to make paper, and it's also used to make threads. Okay, so that's your sclerenchyma. And I know I'm going fast, but you should know all these cell types anyway. Now, if we look at the vascular tissue, remember, vascular tissue is made up of a vascular bundle. All right? And vascular bundle is made up of the xylem and of the phloem and of the cambium. Now, within that vascular bundle, it's going to always form as a ring all the way around with the little sclerenchyma cap on the top. This I just put in so that you remember. The xylem is always on the inside. Okay? Remember in the root, the xylem made that X shape. Okay? And you had your phloem over there like that. That was in the root. All right? Now if we look at the stem, what are we going to have? We're going to have the little vascular bundles all the way around, arranged in a neat little circle all the way around. We have the sclerenchyma cap, and then we have the phloem, and then we have the cambium, and then we have the xylem in the middle, on the inside. Okay? And then your cambium in between the xylem and the phloem. Why? We've gone through this. It is meristematic tissue, and it's that meristematic tissue. If this is the cambium there, and it keeps making new phloem cells, and it keeps, if this is the outside, and it keeps making new xylem cells, it pushes the xylem inwards, and it pushes the phloem outwards. Okay, but it's because the cambium is meristematic. It makes new xylem and it makes new phloem. Okay? This is a micrograph, and the reason I keep putting micrographs in is because some schools don't have micrographs, some schools don't have microscopes. But very often, you wouldn't be able to see this under a microscope anyway, because the school microscopes can't magnify enough for you to be able to look at the structure of a very thin, and to be able to slice the slide or the piece so cleanly and so small that you can actually see it. They use very specialized machines to do this. You've got to know cell structure, and this is the best way. I mean, if the teachers give you this in black and white as a photo and say, here is a micrograph, calculate, have a look. What is this? What is that? Take this and draw a line diagram of what you see. You've got to be able to do that. Now, you'll recognize that this is the micrograph from right in the beginning when we started, and I wanted to show you what the stem looked like. So here are your vascular bundles. They're all arranged in a circle, very close to the outside of the stem. Here is the pith. The pith is right in the center. Here are all your vascular bundles. Now, there is the cambium in each one, and I need to show this to you because it's very important. Well, there's one there, and here's another one there. So, you've got this cambium. That cambium we call, and I haven't really got, so I'm going to try and squash it in here because otherwise you can't see it. It's in, this is your fascicular cambium. Okay? Because it's inside here. Right? And if it's in between, it is, so the cambium, there's still a cambium chain that runs there, and I'm going to color this in dark. So we'll do it like that. There's another one there. Okay? That there is your intervascular, this should be a V, sorry, vesicular, and this is your intervascular cambium. Okay, it's between the vesicular cambium, and there it is inside the little structures. All right. And here's just one single little structure. What have we got? We might as well try green. I hope green's going to work. Okay, there. That is your sclerenchyma cap. Okay, this region here is the phloem. There is your fascicular cambium, and here is the xylem. And if you get asked in an exam, if this gets given to you, I know I'm being mean now, but it would mean that you actually understood it. I would give this to you in a test, all right? A nice, clear, black and white, clearly printed on photostat paper with your rest of your test, all right? And I would ask you to name them and then draw a line diagram of this little vascular bundle. So you would have to label it vascular bundle, number one. Number two, you would have to also tell me that the cells here that are all around are parenchyma cells. You would have to tell me that sclerenchyma cap, that we have the phloem cells, we have the fascicular cambium, and we have the xylem. Right. And there you go. It would be 7 marks just for that. So, and all you're doing is you're drawing a line diagram of what you see here. Okay. Xylem, very important, people. Please, and I'm going to do this in bright yellow. Okay, what does the xylem do? It carries water and dissolved mineral salts, okay, up the stem. Your phloem carries water and dissolved nutrients up and down the stem. The cambium, what does the cambium do? Cambium helps with secondary thickening. Why? Because it makes more xylem and more phloem, and as it makes more xylem, it pushes the xylem inwards, and more phloem, it pushes the old cells outwards. It's like when your nail grows, okay? As your nail grows, where does it grow from? It doesn't grow from this end, it grows from the middle here. New cells are made, okay? And it pushes the older cells back and back. So the cells that are at the end of your finger are the oldest cells. The cells that are the newest are right where the cells are growing from. And the cambium grows new phloem and new xylem, okay? And then we have the thick cell walls of the xylem and the phloem. Why? Because of the sclerenchyma cells. And what are they doing? They're giving mechanical support and strength. Okay, if it's mechanical, it means it's physical strength and support to the stem. Okay, now we look at the pith. And the pith cells, where are they found? They're right in the center of the stem. I need lines. They're right in the center of the stems. What are they? They're large, they're thin cell-walled parenchyma cells, and they have huge intercellular air spaces. And that's why they sort of look like this. So nice big air spaces. All of this here, air spaces. All right, and why? Look what they do. They store water and they store starch. Why do they store starch? Because starch is osmotically inactive, and a plant will take glucose molecules, group them together to make starch. And starch doesn't affect water, so it doesn't change anything within the plant. And then intercellular air spaces, wherever you see intercellular air spaces, it will always be for gaseous exchange. So people, you see how we repeat ourselves over and over and over again. If you learn the structure of the cells one time, it's done. And the same will apply to your human, to the human anatomy and the plant tissue. I mean the animal tissue. Learn the structure versus function of those cells, and I promise you now, you never have to learn it again until you're in matric. Okay, because you'll know it. This is what the pith looks like. I got a picture. And if you look at this here, can you see the very definite circle and all these cells in here? Let's get another color. All the cells in this region here, they're coarse, and there are lots of air spaces because their function is not for anything other than the air spaces. All right. Okay, now we go to what is important, and that is secondary growth. Now, think of the word primary and secondary. When you were little, okay, you first went to nursery school, and then you went to primary school, okay? And you moved from primary school to secondary school or to high school. But new kids came in, or new learners came into primary school. So they came into grade R or grade 1 as you moved on. And then when you went to varsity, what happened? Those learners that came into grade 1, now they move into high school or secondary school. Now the same thing here, except that you've got the cambium doing the growing. And as the cambium grows, it makes a new phloem cell, and or new layer of phloem cells, they are going to push the older layer up. And then another new layer of phloem cells, and that layer that they've just produced will push up, and the old one will push up even further. So that by the end of it, your phloem cells, your oldest ones, are sitting closest to the outside of the stem, and your youngest ones are right next to the cambium. And the same with the xylem. So here, remember, your cambium is meristematic tissue. And I feel like I keep saying the same thing over and over again. If you know it once, and you know it by now, You never have to learn it. Remember, vesicular cambium, that's the cambium inside the little vascular bundle, okay, is meristematic. And what does meristematic mean? It means to make, it's able to make new cells. It's a growing point. Right, now, how does it do it? Mitosis. Why mitosis? Because remember, with mitosis, we go from, or mitosis is for growth, for repair, and for replacement of cells, and also for reproduction in unicellular organisms. This is not a unicellular organism, it's a multicellular organism. Let's just finish this slide, then we can go for a break. Okay?
You know what, Kathy? You can actually just carry on.
Can we? Oh, okay,
because
cool.
we're enjoying it so much. I swear.
I think what we need to do, learners, is send Katlego your next test, okay? Send it to Learn Extra. They've got an email address as well.
Yes.
Send it to them. Special attention, Katlego. And I will sit with her and the two of us will write your test. Okay. I'll be there to encourage her and tell her how excited
and
we
then
are.
everyone would get like 100% because we're just so on the ball here at
Are
Mindset.
you going to get 100%, my darling?
Yeah, and they will get 100% too. I know you will,
cool.
guys.
And even you know now that cambium is meristematic, which means it undergoes
mitosis.
There we go. You see, people, come on, come on.
It's easy.
Yeah. Shame. I mean, she's got to sit here and listen to all of this, and she's done matric. All right, here we go. Meristematic tissue, it divides by mitosis. Why? It's going to make new xylem, pushing towards the inside, and it's going to make more phloem, pushing towards the outside. Now remember, it's going to form the inner and outer rings, which are going to do or result in secondary thickening. And that gives you your annual rings. Now if we look at wood, okay, and I know every single one of you has seen a tree that has been chopped down. So you look at the tree that's been cut down or chopped down, and if you stand over it like this, you can see all those rings. It looks like someone's taken a dark brown Koki and actually marked specific rings. Now, each of those rings represents a period of time that stem has grown. So when you look at a tree that's like 1,000 years old, okay, we sort of guess because you think, sure, it is huge. It's got a stem this size. Unless, of course, it's a bonsai, because then it can have a stem that size and actually be like 1,000 years old. But the thing's only this big. Okay, but if you cut it in half, and please don't, because it would mean that you'd have to kill that plant, you could actually sit and count all the rings. And each ring represents what happened in spring and summer, and what happened in autumn and winter. So every 2 rings means 1 year. That gives you 1 annual ring. All right, so let's see how this works. Okay, here we have the whole secondary growth. And people, this secondary growth is important. You can probably expect in an exam or a test that your teachers will give you a diagram of secondary growth and ask you to explain what has happened. All right. And they're going to expect you to know your labels. So your vascular steel, all right, and that vascular steel is made up of the sclerenchyma cap, which is there for strength, etc. You're going to have the phloem cells, the fascicular cambium, which is meristematic, and you're going to have the xylem on the inside. All right. And it's going to make a ring around the circumference of the stem. So these are things that they could give you and say, write an essay. So make sure you know it, make sure you understand it. So here we go. We have 2 types of wood, okay? Now think, heartwood, your heart is in the middle here. So your heartwood would be the most special wood. So heartwood, it's inside. And because it's right in the middle, it's going to be the older xylem layers. And what's happened? They've become hard and they don't work anymore. So there is no conducting. They are non-functional and non-conducting wood. But their main function here is going to be support. Okay, it would be like having this incredibly strong center part of the wood, and it's there, and it's right in the middle. And what it does is it makes sure that nothing can move sideways, that it holds that tree upright. Okay, it's like a coat hanger, right in the middle. Okay, so it's heartwood is going to be in the inner part, and it's the old xylem. And it's so hard, it doesn't work anymore. Okay, now, we then get, so there are two types of wood. Heartwood is in the inside, and sapwood. Okay, now, sap is your water plus nutrients that travels up and down a plant. So your sapwood is going to be carried, sap will be carried by the phloem, but your water and nutrients, or mineral salts, from the roots is going to be carried up via the xylem. So it's your functioning xylem tissue that we are looking at when we look at the sapwood. Okay, heartwood, the inside, it's not working anymore, it's the oldest part and it's non-conducting. Sapwood is the outer, younger, and functional xylem. And what is its job? It's to carry water and mineral salts up that tree. Okay? And then your annual rings. Here we have autumn wood and we have spring wood. Okay? Now your autumn wood is the xylem vessels. They're going to, it's your drier seasons. And we're assuming here that the rainy season is during summer. All right? So it's during the drier seasons, and what? They're going to be smaller, they're going to be darker in color, and they're going to be more compact. Whereas when we look at spring wood, which is spring and summer, and you're getting lots of rain, okay, they are going to be larger, the lumens are going to be larger. Why? Because they've got to carry a lot of water from the wet soil. Remember, how does water get into the soil? By means of osmosis. So that water keeps going in. Okay? So in summer, your spring wood, lots of rain, it's got to carry a lot of water to the leaves. So the leaves can grow and flourish and make flowers, and so that flowers can be made and fruit can be made. Okay? In winter, everything sort of slows down. So annual wood, your autumn wood, hard, your spring wood, all nice, and it's much lighter. And I really want to show this to you. Okay, we'll put those diagrams on, but I want to just, I've got very little time here. There. If we look here, there are your annual rings. This here is your heartwood. And this here is going to be your sapwood. All right. And that's how your plants grow. Okay? Secondary thickening, make sure that you learn this, make sure you know your diagrams. We are posting them on for you.
Yes, guys, there are some diagrams on the show, on the page. So make sure that you learn more, learn extra, guys. Come on, stay tuned. Grade 10, see you next week for another fantastic life science show. Bye-bye.